Bile does not have any enzyme, it contains bile pigments, bile salts, phospholipids and cholesterol. So what is the function of bile? It helps in emulsification. Very important. Emulsification of what? Emulsification of fats. So this is the fat. We all consume fat, correct? So this fat, when it combines with bile salt, it is going to form a structure like this. So what do you see here? The fat is covered by what? Bile. What is that? Bile salt. Okay. Now this bile salt covered fat is ready for digestion. Okay, or you can see emulsification is a process where the fat is made ready for digestion. Okay, without emulsification the fat cannot be digested. Okay, this is called as emulsified fat droplets. What is the use of this? It provides large surface area for the action of what? Lipase. What is lipase? Lipid digesting enzymes are called lipase. Okay, did you all understand what is the role of bile? Bile helps in emulsification of fats. So you see, while preparing this cake and all, no, we continuously beat it, right? So when you beat, you see a kind of droplets that is coming out, right? So that is also a process of emulsification. Okay, and while they do painting also, right? So they stir it continuously. Why? Because they are preparing the paint for painting. Okay, I am just giving you an analogy. Okay, here we are preparing the fat for digestion. Who is preparing? Bile is preparing. Okay, where is bile produced? Where is bile produced? Liver. Where is it stored? Very good. Bile is produced in the liver. It is stored in the gallbladder. In yesterday's class we discussed that. Can we continue now? Okay, write down. So this picture helps in, see, it has the bile salt has hydrophobic and hydrophilic. Can anyone tell me what is hydrophobic? Water hating, correct? What is hydrophilic? Water loving. So fat is hydrophilic or hydrophobic? Fat, hydrophobic. Okay, so you can see this yellow color, hydrophobic, goes and binds to the hydrophobic region of the fat. So that is about bile. Okay. Next we will see about role of pancreatic juice. Okay. So in yesterday's class we discussed, no, what all does the pancreas, write down, I will give you time, write down. Done? Okay. So in last class we discussed about pancreas, right? So pancreas produces what kind of juice? Pancreatic juice. What all does the pancreatic juice contain? It contains pancreatic amylase, correct? It contains lipase, then it contains three important proteases: chymotrypsinogen, trypsin and trypsinogen and carboxypeptidase. What are the three enzymes, three proteases? Chymotrypsinogen, trypsinogen, procarboxypeptidase. These are all zymogens. Okay. So first see, there are two amylases in your digestive system. One is salivary amylase. It is also called ptyalin. In yesterday's class we discussed, correct? There is one more salivary, not salivary amylase, pancreatic amylase. It is called amylopsin. Okay. What is the name for salivary amylase? Ptyalin. What is the name for pancreatic amylase? Amylopsin. So what does it do? The same thing. It converts starch into maltose, isomaltose and dextrins. Okay. Maltose and isomaltose are disaccharides, dextrin is a monosaccharide, starch is a polysaccharide. So what is digestion? Breaking down of a larger molecule into smaller molecules. Clear? All of you understood what I am trying to explain? Okay. So that is the function of pancreatic amylase. What is the important question they can ask here? What is the other name for pancreatic amylase? Amylopsin. Okay. What is the other name for salivary amylase? ptyalin. Don't forget, when I repeat things, it's very, very important. So, you can see here this duct. So, this duct is going to conduct all the juices into the duodenum. What are the juices? Pancreatic juice and bile juice, and intestinal juice will already be there. Okay. So, next let us try to understand the proteases. Okay. So, which are the proteases that are present in intestine? Just now I told trypsinogen, chymotrypsinogen and procarboxypeptidase. Clear? All of you understood? Okay. Can I continue? What are the proteases that are present in stomach? In stomach, pepsin and renin. Okay, or you can tell pepsinogen and prorenin. Okay, if you want, you can write it down. In stomach, which are the proteases? Pepsin and renin. Okay, in pancreas, which are the proteases? Yes, trypsin, chymotrypsin. Many people get confused, that is why I am making you write here. Chymotrypsin and carboxypeptidase. What do you mean by proteases? First of all, protein digesting enzymes. Done? Okay. So, now let us try to understand the action of pancreatic proteases. Okay. So, what did we study in pancreas now? We studied the digestion of carbohydrate. Okay. Now, we will study the digestion of proteins. Okay. So, you have a hormone, okay, that is called enterokinin. Okay. This enterokinin will convert trypsinogen into trypsin. Okay, and this what is this is all what do we call these as? Zymogens or precursors. Okay, these three are called zymogen. Zymogen means what? Inactive forms. Very good. Then chymotrypsinogen in presence of trypsin forms chymotrypsin. Procarboxypeptidase in presence of trypsin forms carboxypeptidase. Now all the proteases are active. Correct? So, you can see the proteins, peptones and proteases are acted upon by what? All the proteases. Which are the proteases? Trypsin, chymotrypsin, carboxypeptidase, and it is converted into dipeptides. What do you mean by dipeptides? They contain 2 amino acid like this. Okay, this is amino acid 1, this is amino acid 2. I think all of you know that proteins are made up of amino acid. And what is the bond that is present between the protein amino acids? Very good, peptide bond. Okay, next the fats are converted into diglycerides. So, fats are usually triglycerides, correct? So, what is the enzyme here? Lipase. Okay. Pancreatic lipase. There is lipase present in your stomach also, that is called gastric lipase. Okay. Then you have lipase converting into monoglyceride. Okay. Then you have your nucleic acid. In presence of nucleases, it will become nucleotides. Okay. Is it clear? Make a note of all these reactions, very, very important. If you have any confusion anywhere, please ask. And you should also remember enterokinin. Where is it present? It is present in the intestinal juice. So, enterokinin present in the intestinal juice activates trypsinogen into trypsin. Now, trypsin is in turn going to activate your chymotrypsinogen and procarboxypeptidase. So, what is this activation I am talking about? Just give me a minute, I will just explain you this and give you time to write. See, when I talk about activation, Imagine this is pepsin, it will be very long. Okay. So, you have trypsin, this is trypsinogen, this is an inactive form. Okay. Now, this in presence of enterokinin will form trypsin. Okay. What is this trypsin? Trypsin will be a very short version. Okay. There will be cleavage of the peptide, that is called as activation. Okay. So, enterokinin results in activation of trypsinogen into trypsin. All of you got it? Any doubts? Okay. Now, write this down. Next, we will be discussing about action of intestinal juice. Done?